do slide share. Or do you think this is fine that I keep going like this?
If we continue like this, your content will be visible.
Okay, so should I send it to you? Like, should I send you the
Ma'am,
PPT?
you have my mail ID. I'll just message my
Yes.
mail ID to you.
Yes. Mhm. Okay, so we are back. Everybody can hear me?
Yeah, ma'am, you're audible, ma'am.
Okay. So sorry for the technical glitch that we faced, but yeah, I hope all of you are going to enjoy this talk.
A very good afternoon to everyone, and welcome to today's international webinar on model organisms for research related to human diseases, hosted by the PG Department of Biotechnology, Dwarka Das Govardhan Das Vaishnav College. It's not what you achieve, it's what you overcome. It's what defines your career. The best way to predict the future is to create it. I, Kirtana of First MSc Biotechnology, extend a delightful welcome to all the participants for today's international webinar on model organisms for research related to human diseases. On behalf of the PG Department of Biotechnology, I feel immense pleasure to welcome the management, Secretary Sri Ashok Kumar Mundraji, Principal Dr. S. Santosh Babu for our today's webinar. I would like to welcome Dr. M.D. Balakumaran, Head in Charge and Assistant Professor, PG Department of Biotechnology, DDGD, Vaishnav College. I would like to extend my pleasant welcome to the presenter of today's session, Dr. Khairun Nisaa, Postdoctoral Fellow, Ben-Gurion University of the Negev, Israel. I would also like to take this opportunity to express my gratitude for today's speaker, Dr. Khairun Nisaa ma'am, for accepting our invite and joining us for the webinar. I welcome you, ma'am. I would like to extend my warmest welcome to all the heads, faculty members, students, and research scholars of DDGD Vaishnav College and various other institutes. I ensure that this webinar will definitely provide valuable information, and the next 1 hour will be effective for every one of you. Thank you. Once again, I welcome you all for this virtual webinar. I take immense pleasure introducing our speaker to you guys. Teresa ma'am has completed her Bachelor of Science Honours in University of New Delhi, Master of Science in Indian Institute of Science Education and Research in Kolkata. She has completed her Doctor of Philosophy in Ben-Gurion University of the Negev, Israel, and is currently in the short-term postdoctoral position in Ben-Gurion University of the Negev, Israel. Her research experiences include the topics cloning, expression, and molecular characterization of HCP, the functional subunit of T6 secretion system of Campylobacter jejuni isolated from human clinical samples, and the topic proteomic analysis, identification of antigenic proteins in exosomes isolated from Mycobacterium tuberculosis, infected macrophage cell lines. And her recent research is on the topic role of differentiation transcription factor HLH1 in muscle and proteostatics maintenance during Cyanohepatitis elegans larval development. She has a fellowship. She has got a fellowship of Elite Fellowship by the University of Delhi in 2012, IISER K Fellowship by Indian Institute of Science, Education and Research in 2013 to 17, Craitman School of Advancement Graduate Studies by Ben-Gurion University of the Negev from 2018 and ongoing. Now it's time for us to listen to our speaker, Khairun Nisaa. If you have any questions during the presentation, please type them in the chat box and the speaker will answer the questions at the end of the session. Also, this webinar is being streamed live in YouTube. Those who have queries can also post their queries in the chat box in the live session. Over to you, ma'am.
Thank you, Kirti. Thank you very much for the beautiful introduction and a well-written about my career, research career. Thank you. And thank you all for inviting me as a speaker. And this is my first experience as a guest speaker because I completed my postdoc. I completed my PhD recently a month ago, and now I'm currently working with Professor Anat Ben-Zvi in Ben-Gurion University, and we are trying to understand how tissue-specific chaperone molecules are how tissue-specific transcription factors are helping establish the tissue-specific chaperone networks. So, but I'm here to talk about how model organisms help in research and development of human health. So, can I change the slide? Yeah. So, as we all know, what model organisms do are model organisms help us understand the different biological processes, including development of an organism, signal transduction of various signaling molecules. They also help us understand different biological processes such as metabolism and aging and various cellular processes and such as cell division and many other cellular processes which we will discuss further. Okay, so what are the characters? What are the major characters of a model organism? The first thing that comes to our mind is that they should be easy to grow and maintain, meaning they should be in a size that is easy to handle in a laboratory conditions, and they should be able to grow in a laboratory conditions. Second comes the short lifespan. So what comes this means that they should have a it's easy for us to study a new model organism if they have a short lifespan, meaning they should like hatch, they should complete their reproductive cycle, and they should go through all the different developmental stages as much as possible, similar to humans or similar to what we are studying. So we should have model organisms with specific processes that we are looking at. Let's say if we are looking at developmental processes, the model organism uh, should go through all the developmental processes similar to humans. They should be able to produce a large offspring, which means um, it is a plus point, it is a positive point if they are able to produce a large number of offsprings which are genetically similar. You'll see in case of some model organisms that are able to produce hundreds of eggs and hundreds of offsprings which are genetically similar, meaning they have a very similar genome in the genome. This enables us to understand and study a specific process in a large number of samples, which is a mandatory requirement to prove and understand any biological process. Finally, okay, so there is another point called resemblance with human genes. So this is one major character which we need in a model organism, which is how much similar their genome is to human genome. So we'll see that there are model organisms which are not very similar, such as bacteria or yeast, but they have some specific genes which are similar to human genomes, such as that are specifically involved in one or two processes, such as cell cycle, cell division, similar to that, or mitochondrial pathways, something like that. But the maximum number of genes that are resembling to the genes that are present in humans will be considered the most close and most useful model organism, and they should be sustainable nutrient conditions, meaning they should be able to we should be able to feed them a qualitative and quantitative amount of food. For example, if you take an elephant as a model organism, of course you cannot keep it in laboratory conditions. That you will have to have at least 100 to 200 elephants for your research, but it's not possible. And on the other hand, if you try to feed them, I don't know how much you will have to feed them. So that becomes a restricted conditions, and that is not a suitable model organism. So next, I'll jump into. Can you go for the next slide? Yeah. So I'll take a few examples of model organisms. I'll discuss a few of them. So the first that comes to my mind is the Saccharomyces cerevisiae, which is a baker's yeast. Click. Okay. Yes, the baker's yeast. The second one comes to my mind is the laboratory mouse, which are used for various drug development, which are used to study behavioral patterns, which are used to study neurobiology and how neurons and drugs are linked to each other. And the another one that comes to my mind is Arabidopsis thaliana, which is a plant molecular model organism. And you can see how the plant model organism helps us sustain life, because we study a lot of environmental conditions, stress conditions, drought conditions in plant model organisms, which is Arabidopsis thaliana. And finally, the my favorite one is C. elegans, which is called Cinnahabditis elegans. It is a transparent nematode. It's a tiny, free-living, transparent nematode that helps to understand various neurobiological molecular processes such as development, proteostasis, aging, neurobiology, and various molecular and biochemical assays. Can you go next? Okay, so let's start let's start with the yeast as a model system to study cancer. So so cancer, as we all know, is abnormal cell growth that happens due to uncontrolled cell division, uncontrolled activation of genes that are involved in cell cycle. So let's say there are 3 genes in cell cycle. So I will briefly explain uh, how scientists discovered what genes are involved in cell cycle and how are they named. So let's say there are 3 genes that are helpful in cell cycle, cell division in yeast. So they named it as A, B, and C. I simplified it because it will be easy for us to understand. So the cell division in yeast shows that the yeast cells first they grow in size. If you can go to the next slide, they grow in size. Let's say that function, the first function, the growth in size, and then they start budding. They start forming buds, which is initiated by a gene called gene A. Let's say, and then there is a gene B, which actually helps the small bud to grow into equal size as a parent. And then there is another final gene, which is called gene C, that helps the final separation of 2 yeast cells. So the scientists, what they did was they created 3 strains back. They created 3 strains of yeast cells with mutation only in gene A, and then in gene B, and in gene C. So there was a strain with gene A mutation, there was another strain with gene B mutation, and the third strain was with gene C mutation. So they let the uh, yeast strain grow, and they observed under the microscope. So in the process where in the strain where there is, so under normal conditions, yes, next slide. Under normal conditions, what happens is the yeast cells look like this in 0 hour, and in the first hour they started to form buds, and they are starting to form more buds in 3 hours, and then finally they are all separated and they all look different. They are all of the same size, but they are all clumped together. And so when we mutate, when we study mutation, what happens is, yeah, next slide. Yeah. So let's say in a strain where gene A is mutated, meaning gene A is going to is mutated. So the only product that you will see under the microscope is the first stage of yeast. That is only one cell, and they are not able to form the bud, and they're not able to form further separation, and they're not able to produce more yeast cells. So you will see that only one cell of the yeast is observed under the microscope even after few hours of growth. But when gene B is mutated, you will see that gene A is functional. So they will form yeast buds, but they are small in size and they are not growing in size as a parent cell. So you'll see that you observe the yeast with a big parent cell and a small bud cell, because it's not able to grow because of the mutation in gene B. So finally, when you have a mutation in gene C, what you see is you have equal sized um, yeast cells, but they're not able to, separate. That is the final step of forming an individual yeast cell. So what happens is, so this is how they understood that what are the genes and how are they organized in a genome. So first, the functional gene is gene A, and which forms this, which leads to a small bud but not the separation. Gene B, which leads to equal sizing of both the parent and the bud cells. And finally, the C gene, which helps in separation of each of the yeast cells. So each of this gene acts in a clockwise circle, meaning one after the other, and thus they identified that this gene A is involved in budding, gene B is involved in elongation of the bud, and gene C is involved in the final process of separation. So this is exactly how yeah if you can go to the next slide. This is exactly how they looked in the first control is a strain which do not have any mutations in the background. So when that happens, so they are growing fast, they are finally, like, they are growing like wild type genes, wild type is, and they're not producing, or they're not halted at any stage of cell cycle. But when you see in the 369D1 strain, which is a strain with gene A mutation, you see that they are arrested in a single cell stage, meaning they're not able to form the gene. They're not able to form the they're not able to form the bud in case of mutation in gene A. And then in 370D1, which is a mutation in gene B, so they are able to form the bud, but they are, since they are not able to, they're not able to grow in size. Finally, in the 104D strain, which is a strain with mutation in gene C, you see that they are still growing in size and in number, but they are not able to separate, because the function of gene C was to separate the cells, the daughter cells from the parent cells. So this is how they identified. If you can go to the next slide. So if this is how they identified that there is a gene called, the gene A was identified as CDC1, which is a cell division control gene 1, and gene B was control CDC2, and gene 3 was CDC3 gene. So this is how they identified what are the genes that are regulating the cell cycle. So this information can be exploited, and this information can be put into use to understand how cell cycle genes are regulating cellular division. and thus can be targeted for therapeutic use by cancer therapy, for cancer therapy. For example, there are many drugs that are targeting the mm, targeting and inhibiting specifically the CDC1 gene. There are drugs that target CDC2 gene, just that they will form they will form two different cells, but they won't be able to separate. And also, so this way you can identify genes in different model organisms and can find their homologous genes in human processes, cell division or developmental processes. And you can also identify similar genes in aging, health, and any process, development, and everything. So this is one very simple and very clear example of how model organisms and their genes can help us identify similar genes in humans, and they can be finally targeted for therapeutic studies. Okay, so next we can move on to model organism, which is C. elegans. That is my favorite because I worked throughout my PhD in that. And so, a little brief introduction. Sidney Brenner was the first scientist who made C. elegans, who isolated the C. elegans from free soil, and he grew them in the laboratory conditions. He is the one who uh, kind of formulated the nutrients that are required for them, and he is the one who started culturing them under laboratory conditions. So it was in 1963, and finally he was awarded a Nobel Prize in 2002 for his discovery and for his studies with neurobiology. So what are the features of C. elegans, let's say? So they are hermaphrodites, meaning they have both the male and the female gametes. So they are able to produce, they do not need males to reproduce. So one female, like one single organism, one single hermaphrodite is enough to generate 200 to 300 offsprings. So meaning, if you are left with only one organism of that strain, you can still recover the whole strain for your research and studies. Yeah. The second thing is they are transparent. So this makes a lot easier for us to observe under the microscope, a lot of different processes, such as how the cells are growing and dividing, how they are forming um, all their organs, and it's easy for us to study and look under the microscope and see, because they are transparent, because they are you do not require dissection, or you do not require complicated processes like this. They are cheap to maintain, meaning they eat the bacteria, so you do not have to make special chemical nutrients in your plate, or you do not have to worry about how you're going to feed such a large number of C. elegans in your laboratories, because they eat bacteria. So, and bacteria grow very fast, and you can feed them. They are also very easy to give heat shock, or to study different cell conditions, different developmental processes, because you will see that they have a very elaborate cell life cycle. So yeah, and then finally they are very, very resilient, meaning they can withstand heat shock, cold shock. They can starve for a few months. They can just survive without food for a few months. And there are different interesting pathways that are activated when they are starving. Okay, so let's check their life cycles. Can you go for the next slide? Hello, Kiran. Hi.
1 second. Ma'am, I think it might be a network issue, ma'am. The child has been
Okay.
changed, but it's not visible.
Okay. Maybe I should, go on with the type of slides that I have. Maybe it is still okay.
Yeah, ma'am. Okay, ma'am.
Yeah. Yeah. It's better because.
Yeah, no issues, ma'am. You can.
Okay. So we're here. Okay. Okay. Is it like? Can you all see it?
Yeah, ma'am. It's visible.
Okay. So one of few of the best features for the C. elegans nematode is they have a very short lifespan. They hatch from the egg, they reproduce, they hatch from the egg, they go into different larval stages. They produce, they start reproducing, and then they become adults, and then they age for a few days, and then they die. So it all comes under a few months, meaning, so from the time they are hatched until they become adults, it's only 3 days or 3.5 days, depending on the temperatures that we grow in. But they are comparatively have very short lifespan. So this helps us to study, let's say, if there are genes that are activated only in the late stage of our life. So, or they go, if you accumulate some mutations, and that mutation is only uh, you know um, uh, that mutation specifically only produces a functionally mutant protein, or the effect of that mutation is only visible in later in life. Let's say that type of genes can be studied in this organism, because they have their whole life in within 1 month, and you can study that gene. Otherwise you will need 60 years to study in a human. So, okay, so they have very small size. They are 1 millimeter in size. You'll have to see them under the microscope, and they are, and it's this is one of the reasons why they can be grown in a small plate, or like a agar plate. You all must know, like agar plate, and you can get hundreds of mm, offsprings in that plate. And the one of the major and important aspect is that 59% of the C. elegans genome have similar genes in humans. So there are genes that are functional in the neurons. There are genes that are functional in muscles. There are genes that are functional in intestine. So all of those genes are also found in the C. elegans, and very similar, like 70% of their homology. Like, let's say if there is a gene in humans that makes muscles, is very similar, like 70% similar to the gene that makes muscle in the C. elegans. So this is one very useful point. Okay, so they have fully sequenced genome, meaning the whole genome, like the whole DNA, is sequenced. We know exactly where which genes are expressed. We know which genes are after what genes, and how they are transcribed in a C. elegans. They produce a large number of genetically identical offspring. So this comes from the point that they are hermaphrodites. They produce, so they have eggs, they have sperms inside them, and they can produce as many offsprings as they want until 5 days of their reproductive health. So what happens is, since there is a single parent producing a large number of offspring, they are genetically similar. So they also have 40% of genes that are associated to be with the human diseases. So 40% of the genes that are associated with human diseases are also found in C. elegans. Finally, one of the major and a beautiful technique is genetic genetic manipulation which happens in which we can do in C. elegans. So as you can see here in the, these are the germ cells for the C. elegans. So you can inject the plasmids with gene mutations. You can inject your own DNA, like your desired gene or your desired DNA, and that DNA will be integrated into their genome. Which means, if you, let's say, maybe you some of you might know that there is a protein called GFP, which is a green fluorescent protein, and that if they have that has a specific, like a very short DNA sequence. So if you take that DNA and inject into the germ cells of the C. elegans, what happens is, most of the cells will take up that green fluorescent protein DNA, and they will integrate into their genome. So whenever in all the cells that take up the GFP DNA are going to glow, and they are going to be identified as the cells that took up the GNA from outside. So this will confirm that you can manipulate the genetic background. You can introduce some mutations, you can delete some genes, you can knock down, knock off. All this process is easy in C. elegans, and you can see under the microscope while doing it. So this makes it is a very beautiful and interesting model organism. And another point that I did not include, but it comes to my mind, is they can be freezed in minus 80 degrees, and or using a liquid nitrogen, and they can be used after 20 years or 10 years of use, and they will have the same genetic background. So let's look at how the C. elegans develop and what is this life cycle. So from the stage they are laid, meaning they are embryo, from the two cell stage, it takes around 14 hours for them to become the first larval stage. The first larval stage is around 0.25 millimeter in size, and so they are grown in in a plate with bacteria. So once they have the food, they enter into larval 2 after 12 hours, and their development continues as a normal development, and they reach larval 4 and larval adulthood around in 3.5 days. Uh, but if you do not give them food at L1, what happens is they kind of arrest themselves in the larval 1 stage, and they enter into a state called dower larva, which is a very interesting um, phenomena, and they can survive here for several months in this dower stage, and they return to larval 4 stage when food is provided, when the favorable conditions are provided. So it is a very interesting path to study how C. elegans, which have so many similarities with the humans, can enter into such dower stage and can survive there for few months, and finally can also reproduce and make more C. elegans at the same time when the favorable conditions arrive. So this is one of the interesting research topics that is going on, and scientists are trying to find out what genes are activated during this pathway, what genes are shut down during this pathway, and what happens to the reproductive system, why they are kind of shutting down, kind of hibernating at this stage, and how are they able to survive without food for a few months. So this is all about its life cycle. So let's move on to a few of the interesting anatomy parts, which is the C. elegans have a very clear nerve ring, which is similar to brain cells or brain itself in humans. They have a very clear and um, very clear the body wall muscle cells, which are homologous to human skeletal muscle cells, and they look also very similar. They have all the myosin, actin, sarcomeric structures that are similar to human muscle cells. They have dorsal and ventral nerve cord, and they also have spermatheca and oocyte pouches. They have gonads, they have embryos, they have everything required for this tiny organism to survive. And they also have male in their population, but they are produced very less in number, which is only 0.05 percentage in their population. So okay, so this is cross section, and this is where exactly how the body wall muscle cells are located in both the dorsal and the ventral side, and they run along the length of the body. You can see that it start from almost before the nerve ring, and they are present throughout the body until the tail region, also in the ventral sides. So this helps the C. elegans to move, um, like the skeletal muscle helps us to move and run and do all other activities. You can probably see how they look under the microscope if it plays well. I'm sure. Yeah. This is how the C. elegans look under the microscope, and you can see all these things are the bacteria that they eat, and you can see different sizes of C. elegans, and you will see that these are the embryos that are laid on the plate on the food, and this is exactly a larval 3 stage of the C. elegans. So the sizes differ in different larval stages, so you can identify them at what stage they are. Okay. So since they are very tiny and their whole genome is sequenced, they have a very specific time points of cell division. So it is well established that approximately in 300 minutes after fertilization, the first muscle gene starts to express. So you can appreciate the intricate details that the scientists have studied using the C. elegans. At approximately 300 minutes right after this fertilization, the first muscle gene is expressed, meaning that the muscle structures are formed, the sarcomeric proteins are assembled, the muscle starts to move. And by 450 minutes of fertilization, all of the muscle structures are formed, and they can they show some kind of movement in this threefold stage. So this is still inside the embryo, but they are completely formed. So this is so the body wall muscle cells. This is what I worked with. So I studied how similar they are to skeletal muscle cells, and why they can be used to understand neurodegeneration and muscular dystrophies in humans. So, to understand that, we need to know what a proteostasis network is. The proteostasis network consists of 3 modules, which are protein synthesis, protein folding, and protein degradation. So, all of these 3 modules have to be interlinked. One has to, there should be a balance between all 3 of these processes. Otherwise, there will be a mess inside the cell. So one of the protein that helps to regulate and maintain the whole cellular integrity is the molecular chaperones that are helping in folding and refolding of proteins. They also help in the degradation of proteins. They also tag the proteins that are misfolded. They also tag the proteins for sending it to the different cellular locations. So these molecular chaperones are regulated by a gene called HLH1, which is what I studied. So I'm not going to go into the details of my research, but I'm just going to, give you a background. So HLH1 is also exactly the homologue of MyoD. MyoD is the gene that makes the skeletal muscle cells in humans. And this is exactly similar to the gene that we have in C. elegans, which is HLH1. So we wanted to study what happens if the HLH1 is mutated, or if what happens to the developmental processes of body wall muscle cells in C. elegans, what happens to the movement, what happens to the body structure, the size, the shape, all of their phenotype. So what we understood is we use a technique called thrashing assay to understand and to study and to observe how fast the animals can move in a liquid media. So this is a representation of how the worms move, but it's not exactly what we do with the worms. So you can see that the wild type are the animals that are control animals, that they do not have any mutation in the background. So they grow into 1 millimeter size, they are fully functional, they are fully motile, and they grow, they at different conditions, they are all having approximately 200 times, faster they move compared to the mutation with compared to the worms that had mutation in the the gene, that is HLH1. So you can see a clear difference that this is how big the wild type worms look. On the other hand, the worms with HLH1 mutation, they look very stunted in growth. They do not move. They do not reproduce as much as the wild type. They also have very um, disorganized muscle structure. So this is how their muscle structure looks in a wild type gene. They are very crisp. They are very clear. They're very straight lines. But if you look at the mutation, if you look at the mutant worms that make the muscle cells in C. elegans, you see that due to the mutation, all the myofilament organization is disrupted, and this is why they do not grow into full size. They do not move faster. They do not even move at some cases. So so this is how we kind of extrapolate the results into human studies. So let's say if HLH1 is mutated and we see such a severe phenotype in motility, in growth, in reproduction, all of it, so it can be possible that it can be applied to human muscle genes, the MyoD, and understand what happens there if you mutate MyoD genes. So this is one example of what I studied and what I understood from using a model organism, how you can relate the processes, the cellular organizations, and different developmental processes, and you can extrapolate that result and understand human physiology, human metabolism, human development. So another example of C. elegans as a model system is the use of C. elegans for Alzheimer's disease. So Alzheimer's disease is a late onsetting disease, meaning you will not know when the Alzheimer disease starts to, show some effects or show some symptoms. So it is a gradual process. So what happens is there are different type of proteins that are, for example, like one of the major known protein is the tau protein that starts to denature, and that starts to misfold, and then they starts to aggregate, like they form aggregates. So in a normal brain, this is how the space and the volume of a normal brain looks. But on the other hand, this is how Alzheimer's brain looks. So why C. elegans is a good organism to study Alzheimer's is because they have very similar brain structure called nerve cord, and they have approximately 1 month or 2 months of their lifespan, which is equal to 60 to 70 years of a lifespan of a human being. So what whatever you can understand and study using human as a model organism in 60 years, you can do it in a C. elegans in 1 month or 2 months. So that gives you, and you can repeat as many times as you want. You can use there are like the number of samples can be increased, the number of mutations can be increased, the number of different combinations of mutations can be increased. So this is how a normal brain looks, and this is how a normal neuron looks. On the other hand, the Alzheimer's brain looks like, you can see that the amyloid plaques are being formed. They deposit between the neurons, and they block the signal transduction between the neurons. So this happens approximately later in life, and you might not have clinical samples to completely study the process. So you can use C. elegans to understand how deposition starts to occur, when it starts to occur, and how can you use a drug or something to dissolve this kind of the plaques, the amyloid plaques or fibrillar plaques. So yeah, so this is how the C. elegans. Let's say there is a tau protein. You can put that gene, like you can introduce that gene. Like I told you, you can inject that specific DNA sequence into C. elegans, and you can overexpress them. You can you can overexpress that specific gene, and then you will see that this amyloid plaques are formed due to the overexpression of human tau protein. So when you overexpress the same tau protein in the C. elegans, what happens to their neuronal transduction, like how the neurons are affected to signal the other neurons, how they are able to like what are the behavioral differences when there is a plaque in the nerve ring, what happens, and what happens to the other organs. Let's say what happens to the muscle structure, what happens to the intestine, what happens to other reproductive system. So all of it can be studied using this C. elegans, and you can easily genetically manipulate them. You can overexpress them. You can create a mutants of that gene. You can use some drugs to treat C. elegans, and you can see what happens if you treat the same drug to humans. Obviously, you cannot directly take it from the lab to but treating a patient. It takes a long path to reach that stage. It requires clinical trials. It requires a lot of research, further research and confirmations. But this can be an initiation point to start with a lead drug. Okay, another aspect is aging. So aging is a natural process, we all know, but aging healthy is the goal of the scientist. So what happens is you age, but what happens during you age, you all the other signaling pathways such as insulin pathway, the germline signaling, why does the reproductive system uh, reproductive capacity reduces, why does it happen during age, and what happens to the insulin signaling, why do we all start to develop diabetes or other metabolic disorders. So all these pathways can be studied in C. elegans because they have a very, very similar and closely related signaling pathways in humans. So, and then you can use other studies such as energy metabolism. You can study how fat cells deposit, how do not deposit. You can also study what happens to the immune system of C. elegans, and then you can extrapolate that result to or predict, oh, this is something that might be happening in humans also. So you can look for a specific gene. Let's say if you identify a gene that goes that gets mutated over time and specific mutation accumulates, you can look for the same gene in humans and see whether same kind of mutations accumulate, or what happens and why does it accumulate. Why does the DNA repair system not function at a certain age or certain time point? So you can correlate all the findings of your C. elegans, of your model organism, to study and understand human processes in future. And finally, all these processes are involved in aging, and blocking some or overexpressing some can extend lifespan or improve health span. And this is how we are trying to take what we understand from C. elegans or other model organisms to humans and create some um, healthy life span and healthy aging. So okay, so finally, C. elegans, as I know, I am kind of stressing upon it because I love it, and I have experienced how easy it is to work with, and how easy and how many tools are there in the science field to understand C. elegans. So you can have environmental factors. You can treat the C. elegans with high temperatures or cold temperatures, and you can see all the pathways that are affected. You can see different neuronal signaling pathways. You can see how easily they digest food at different temperatures, or how easily they what is the best suitable condition for living, and all the other biological pathways. And you can treat the C. elegans with different chemical compounds, and because they can eat it and they can easily produce results, because all the cells are going to get it, because it's a very tiny organism. And you can study genetic factors. You can mutate different genes. You can study different pathways. You can study epigenetics, because you know exactly which genes are methylated, which genes are phosphorylated. You know everything about C. elegans genome, and what happens over time, or what accumulates over time, can be studied using different tools and techniques which are very prevalent, which are very easily available for C. elegans. Finally, for aging, it's a very short lifespan, and different type of different time points, like development, like embryogenesis is there. There are different larval stages. There are reproductive phases. There are aging phases. So you have different um, stages of life which can be studied for aging purposes, and various other things like autophagy, mitochondrial health. All of it can be studied using model organisms, or most of the model organisms, but C. elegans is for sure. And finally, I would like to thank Professor Anat Menzwi and all of you for listening to me. And I'm up for questions.
Participants, if you have any queries, you can unmute yourself and ask the queries.
Okay. Wait a second. I think I have some. Where do I see?
Participants, I repeat, if you have any queries, you can unmute yourself and ask the queries.
Is there anyone who has any question? They can directly write me an email or contact me through Kiran ma'am, because I am not able to, read any questions here. I don't know. I'm very new to Google Meet.
Thank you, ma'am. If there are any queries, we'll forward it to you.
Okay, thank you very much. Thank
Ma'am,
you,
can I ask you
Kiran
a
and
question, ma'am?
Kirti.
Ma'am, can I ask you something? I saw your PPT and all your graphics were too good. Can I know how you did it, ma'am? Is it your drawing yourself or you did some graphics over there?
Okay. So the CL against graphics, there is a person called Ellie. You can follow her on Twitter. So she is a person who's working with C. elegans, and she is kind of passionate about making cartoons of C. elegans, and she kind of comes out with her own creative mind to make such beautiful cartoons, and I love it. So I use it all my presentations.
Thank you, ma'am. And one of our participants has a query. Can I read it for you, ma'am?
Yes, yes, yes. Go ahead.
What are the complications we'll face when start working with model organisms?
So I have worked with cell culture. I have worked with Drosophila model organism. I have worked with chicken as a chicken as a model organism, and finally I worked with the C. elegans. So some of the major disadvantages of using a model organism is sometimes you do not know when they're going to die. So you might be in the middle of your experiment. You might be using, and you are expected to use the same model organism or the same genetic background for each and every study or each and every experiment you do. So what happens is, let's say in case of mice, you are supposed to keep a very very clean and very uh, uh, free of bacteria, free of infectious things, to keep them alive. And sometimes you will have to maintain them because you will have to take care of them. They are going to be your babies for the research. So you will have to forget about you know holidays. You're going to forget about how you're going to take care of them. You will have to feed them at the right time, at the right quantity, and you cannot say that it's a Sunday, I'm not going to go to the lab. So that can be one of a major challenge to work with a model organism, because you will have to take care of them. You will have to look at them, how they're growing, if they are showing any kind of symptoms or any behavioral patterns that are different from organism that you find initially receive. So you will have to obviously at molecular levels, you're going to get results only after you dissect them, only after you take the cells that you need from them. But on the outside, you should know that if they are looking healthy, they are eating well, they are drinking well. So all that comes like a challenge, because you have to take care of them as your baby. So that's one challenge, but it's interesting. Did I answer your question?
Thank you very much, ma'am. Thank you for answering all our queries. Thank you very much, ma'am, for your amazing presentation. I'm sure
Thank you
all of
very
us have
much.
got to know through it. Uh, Now, Divya from First MSc
Yes.
Biotechnology will come to.
I'm sorry, I did not hear you.
Gratitude. Gratitude is not only the greatest of virtues, but the parent of all others. On behalf of the PG Department of Biotechnology, I, R. Divya of First MNC Biotechnology, take this carefully. Gratitude is not only the greatest of virtues, but the parent of all others. On behalf of the PG Department of Biotechnology, I, R. Divya of First MSc Biotechnology, take this opportunity to propose the vote of thanks to those who have directly and indirectly contributed to this international webinar on model organisms for research related to human diseases, organized by the PG Department of Biotechnology, Dwarka Das, Govardhan Das, Vaishnav College. At the outset, I express my sincere thanks to our Secretary, Sri Ashok Kumar Mundraji, and Principal Dr. S. Santosh Babu sir for supporting us to conduct this webinar. I want to extend our earnest thanks to our guest, Ms. Khairun Nisaa, postdoc doctoral fellow, Ben-Gurion University of the Negev, Israel. Thank you so much, ma'am, for accepting our invitation and giving us such valuable information. I would like to express our generous thanks to our head, Dr. M.D. Balakumaran, for giving us the necessary support for organizing this webinar and our faculty members for their cooperation. Our heartfelt thanks to the participants of PG Department of Biotechnology, Dwaraka Das, Govardhan Das, Vaishnav College, and various colleges for their active participation in this webinar. Once again, we thank you all for being with us. It has remained a pronounced pleasure. I thank all of those who have joined this virtual webinar and made it a great success. Thank you.
Thank you very
Ma'am,
much.
there is one more query. Can you address
Yes,
it, ma'am?
yes. I saw
What are
that.
the criteria?
Yeah. What are the criteria for selecting a new model organism? So I was going to write something about it, but let's discuss it. It's it's a good question. So let's say let's say uh, recently there is a new organism called tardigrade that is gaining the speed to become a model organism. So what happens? Why did people choose tardigrade as a model organism? It lives forever. It lives forever. So if you want to study some processes, or if you want to study or make changes to some process, such as if you want to make human life immortal, so you're going to choose such an organism to study. You're going to choose that organism, like such as tardigrade, which doesn't it doesn't die for hundreds and thousands of years. So you're going to look at that organism and look at what genes are helpful in the life cycle, and what is the difference between that gene and human gene. What can we manipulate? Where can we take that part of gene and insert into a human gene, and then check if they function like they function in the tardigrade, and they can also function in humans. So for example, in C elegans, let's say in C elegans, let's say if I can, let's say I identified a gene HLH1 that is very similar to the muscle gene MyoD. Let's say I want to increase the strength of the muscles in humans without eating any protein or any meat. So what I'll do is I'll try to understand what genes are, what parts of the gene are helping building muscles in C. elegans. I'm going to take and study that gene, that part of the gene, and kind of try to delete it, try to insert some other gene and see if that function can be uh, altered. So if that can be done, it can also be done, possibly done in humans or other model organisms. So this is how you select a model organism. What process do you want to study? There are model organisms that you can study for different processes. Development, for example, development was well studied in chicken cells, because you can observe the chicken cells, how each and every cells are formed under a microscope without just after you take out some parts of the eggshell. So what process do you want to study? You want to study and understand how the cells react to heat stress, how the cells react to cold stress, or how the cells react to different drought conditions or nutrient deficiency. So you'll have to choose an organism that can survive that nutrient deficiency, or the organism that can still live and function without that gene. So how does it do? So it will give us an insight how that model organism, if it is able to do this with the mutation, how does it how can we use that the whole system, or the whole knowledge about what are the other functions of other genes, we can use it in the model, different model organisms or in humans. but it will take, like, none of these mutations, or none of these deletions, or none of the manipulations that we can do in a model organism can be directly done on humans, because it is a huge ethical issue, because humans cannot be used as a model organism for human studies. Humans are, like, there comes an ethical issue. So there are also ethical issues for other model organisms, such as mouse, the chimpanzees, and other things. Like for C. elegans, you don't have an ethical issue, because it's not. I don't know why it's not, but it's very easy to manipulate genes, and they are not causing environmental. What do you say? Like problems to the environment, so they are not. So this is how you can use model organisms to understand different processes and choose your model organism based on what process do you want to study. Okay.
Thank you, ma'am. I once again thank you. Thank you all. Now that marks the end of our session. Participants can fill the feedback form posted in the chat box, and you will receive the e-certificate in a week's time. Participants, kindly fill the feedback form. The link for the feedback form will close in 15 minutes. Thank you.
Thank you. Thank you very much.